Hello students, this is Barkha Gaur from Shiv Jyoti Educational Group Kota. How are you students? I hope all of you are fine at your places. So students, today we are going to start new chapter of biology class 11th. The chapter's name is plant growth and development. Children, I would like to tell you some of the parts of this chapter are deleted. So you open your book, page number. The chapter is plant growth and development and page number is 239. Okay. When we open the chapter, we see different topics like growth, then development, growth rate. Okay. So, different topics are there which are related to growth and development. So, these topics are being removed for this year. So, what we have to study is we have to start from plant growth regulators. So, the topic we are going to study today is plant growth regulators. Now, when we talk about plant growth regulators, we can also call them as hormones. We have hormones in our body which controls the different activities and different reactions of the body. They also affect our growth and development. Okay? But in plants also certain hormones are there. Actually, in plants we should not call them hormones. They are plant growth regulators because they are controlling and regulating their growth and development. So, there are different categories of plant growth regulator and this topic is starting from page number 247. So, all of you open page number 247 and we are going to discuss these plant growth regulators. So, when we talk about plant growth regulators, we see that they are regulated by some chemical substances. That means the growth in plants is regulated by some chemical substances which can move from the site of synthesis of the site of action. जैसे कि हमारी body में hormones are produced by glands and when these glands secrete the hormones, they are used on the other target site. So, there are two locations we can say, from one location where the hormone is produced and the other location where the hormone is required. Another thing, they are required in very small quantities and they are known as, they can be called as hormone and they act as plant growth regulators. Now, we will categorize these hormones. There are 5 plant growth hormones that exist in nature and they are auxins, cytokinins, gibberellins, ethylene and abscisic acid. Now, when we can group them in the sense that some of the hormones are growth promoting and some are growth inhibiting. So, we can say auxins, cytokinins and gibberellins are growth promoting hormones and ethylene and abscisic acid are growth inhibiting. So, we will understand them in detail. First hormone which is we have to study is auxins. Auxins, the meaning of auxin is to grow. Okay. These were discovered. Now, we will what we will do, we will take the hormone, we will study the hormone. First of all, we will study about the discoveries of the related to this hormone, how these hormones were discovered, and then we will understand the physiological effect of these hormones on the plants. So, first of all, we can say it was discovered by Charles Darwin and his son Francis Darwin. They were studying what they were studying? They were studying the response of coleoptiles of canary grass towards light. ठीक है, ये स्टडी कर रहे थे कि ग्रास में कोलेप्टाइल्स का क्या रिस्पॉन्स रहता है. They observed that the tips of coleoptiles bent towards the light. उन्होंने देखा कि the tip of the plant were bending towards the light. This phenomenon is also known as phototropism, which we have already studied in class 10th. It was proved later that the tip of coleoptile contains a transmittable substance. That means something is there which causes the bending of the coleoptile. तो वो क्या था जिसकी वजह से bending हुई? The transmittable substance were later isolated by F.W. Wendt from oat coleoptiles. These were named as auxins. So, this was about the discovery of the auxin hormone. Auxin was first isolated from human urine and the chemical name of molecule, that means we can call chemically the auxin is also known as indole 3 acetic acid. So, these are the derivatives of indole 3 acetic acid. Auxin also include, there are certain other substances also present in the auxin, which is like indole butyric acid, naphthalene acetic acid, 2,4-dichlorophenoxy acetic acid, जिसको 2,4-D कहते हैं short में, and it is used as pesticides also, weedicide. They are natural and synthetic group of substance which have growth regulating activity in plants. So we saw that there are different kinds of auxins. Now we will understand the physiological effects of the auxin. That means what are the effects which are being expressed in the plant. तो auxin is produced at the growing tips of root and shoot. तो सबसे पहले हमें पता होना चाहिए where they are produced. They help in the induction of rooting in isolated culture and they promote flowering in some plants. So these are the effects, and they act against other hormone abscisic acid to prevent the abscissin. They also promote abscissin at mature stage and they are responsible for the shape of plant, streamlined shape of the plant. That means they exhibit apical dominance. Now what is apical dominance? आपने देखा होगा, the plant grows in height, ठीक है, उसका जो एक मिडिल स्टेम होता है वो ग्रो करता जाता है, लाइक दिस इट विल ग्रो. मतलब प्लांट अगर एक ही डायरेक्शन में ग्रो होता जा रहा है, दैट मीन्स द एपाइकल एपेक्स ऑफ द प्लांट, दैट मीन्स टिप ऑफ द प्लांट इज ग्रोइंग कंटिन्यूसली. तो एपाइकल डोमिनेंस का मीनिंग ये होता है कि इट विल नॉट अलाउ द लेटरल ब्रांचेस टू ग्रो ऑन द प्लांट. ठीक है, मतलब बस ये एक ही, ओनली वन स्टेम या वन ब्रांच इज ग्रोइंग अपवर्ड्स एंड द अदर लेटरल ब्रांच आर नॉट कमिंग. That is called apical dominance. You can see initiation of rooting in stem cuttings. That means जब हम stem cuttings को grow करवाते हैं तो उसमें rooting can be initiated by the help of auxins. This is strong apical dominance in gymnosperms and this makes the shape of gymnosperms like conical. इससे conical shape में भी help मिलती है. One of the function is the auxin induce parthenocarpy. Now you have already studied about parthenocarpy, means the production of fruit without fertilization. That means no seeds will be produced in the fruit. So these are the example of parthenocarpic fruit, and it can also be used as dicotyledonous weedicides. Weedicides के form में भी हम इसको, जैसे अभी मैंने example आपको बताया था 2,4-D. It can be used to kill dicotyledonous weeds which are harmful for the plants and they help in cell division to some extent. The second hormone which we have to study is gibberellin. It's very interesting. We will study about its discovery. तो ये बहुत ही interesting सी चीज़ है. देखिए, gibberellin, which is another kind of plant growth regulator, it was first observed by Ichi Kurosawa, Japanese है ये, in 1926. Actually, what he was studying the foolish seedling disease of Bacne in rice plants. He saw some of the plants of this rice were increasing in height, बस लेकिन height में तो बहुत increase हो रहे थे, but they were not having seeds inside them. So that's why this disease is called foolish seedling disease. So he was observing this disease in rice plants. Then what happened? This disease was caused. He came to know that this disease was caused due to one fungus which is known as Gibberella fujicuroi. Gibberella fujicuroi. So the rice plant affected with Bacne. This is the figure of the picture of the plant. Affected plants etiolated, that means they grow in height but they produced empty panicles, means they do not have seeds. So what happened due to that? They could not support their own weight. जो plants थे वो अपना weight भी bear करने में incapable थे and they just bent down. They toppled over and died. So they could not survive due to their long height. Another example is also there, the foolish seedling disease as observed in cabbage plant. देखो such a long heighted plants are there but they are of no use. So this disease was developed due to the fungus and the acidic substances. जो जिस acidic substance की वजह से ये disease हो रही थी, that was isolated from sterile filtrate of this fungus and was named as gibberellin or gibberellic acid. तो ये acidic है इसलिए इसका नाम gibberellic acid है. So in this way we came to know that how gibberellin was discovered. Gibberellic acid was one of the first isolated GAs. The first gibberellic acid था GA3. This was the first discovered. Now we'll see the physiological effects of gibberellin. It enhances growth regulators, that means जो growth regulators हैं उनको enhance करेगा. Then it causes growth of internodes. Now you should know what is internode. Internode, suppose this is a stem, you can see some nodes here, है ना? This is one of the node, this is another node. तो ऐसे ही आप जब plant देखते हो तो some branches are arising from the node part, but the part between two nodes के बीच वाला part क्या कहलाता है? Internode. तो इसकी length को increase करने के लिए, to increase the length of the internode, the gibberellin is very helpful. तो it causes growth of internodes. And overdose, अगर हम gibberellin का बहुत ज़्यादा overdose देते हैं तो what will happen? It will increase the height of the plant very much, and those plants will be called as giant plants. Also it is used to increase the length of grape stalks. तो जब it can be used as for increasing the length of the grape stalks. Then they cause fruit to grow in size and attain attractive shape. तो particularly grapes में इनकी large size के लिए they can be used. They can be enhanced for malting and brewing, and by the application of GA in sugar cane, long internodes can be obtained. क्योंकि sugar cane में तो हमें क्या चाहिए? Stem की length ही चाहिए which contains sugar. Then it hastens maturity period in juvenile conifers. तो काफ़ी लंबे समय तक they will remain juvenile, that means they will remain young, and it leads to early seed production, promotes bolting in beet and cabbages, promotes breaking of dormancy and seed germination. Now what is dormancy बच्चों? Dormancy होता है कि suppose when we collect the seed, हमारे पास seed रखे हैं, we store them for a long period, we can store them for a long period. That means हमने जो development है या growth है plant की उस समय we have stopped it. How? Because seed are capable of remaining in a dormant stage. Dormant मतलब होता है inactive stage में, that means now no more growth or development is taking place in that part of the plant. So gibberellin is used for breaking that dormancy. That means it will help in growth and seed germination में भी ये help करेगा. You can see these are the sugarcane plants. Long internodes of sugarcane by application of gibberellic acid makes it a more profitable crop. Similarly, gibberellins can induce germination in such plants जो बहुत short time के लिए viable होते हैं. तो those seeds can be germinated with the help of gibberellin. Now the third hormone is cytokinins. Naturally, cytokinins are present in the regions where cell division takes place very rapidly, like root tip, shoot tip and young fruits. तो यहाँ पे क्या होता है? बहुत ज़्यादा speed में cell division takes place. The site of active cytokinin biosynthesis is cambium. Cambium पढ़ा था हमने जब secondary growth होती है plant की, तो between the xylem and phloem there is a ring like structure which helps in increasing the number of xylem and phloem. So that means it cuts the cell और ये secondary growth, that means it helps in increasing the girth of the plant, thickness of the plant को increase करने में help करता है. तो the site of active cytokinin हमें कहाँ मिलेगा? जहाँ पे cambium present होगा, because it is continuously dividing. These were first isolated by Fox-Skoog. You should remember the name of scientist during 1940s. Cytokinins were first crystallized from autoclaved herring sperm DNA and was named kinetin by Skoog and Miller. Skoog and his co-workers were studying growth in tobacco plants. तो all these discoveries related to these growth hormones जो हैं, ये growth regulators हैं, ये discoveries सब accidental है, means the scientists were doing something else and they they came to know about these hormones. तो they observed a strange phenomena. क्या देखा उन्होंने? Internodal segments of tobacco stems could develop a callus. That means वो internode part में भी a new mass of cells was able to be produced, which was undifferentiated mass of cells on treatment with some specific solutions. The tobacco stem cuttings were grown in artificial culture medium. तो इनको यहाँ पे हमने grow करवाया. For in vitro callus formation. In addition to auxin, the medium was supplemented with, मीडियम में और हमने कुछ चीज़ें ऐड की, that is extracts of vascular stems, coconut milk and yeast extract or DNA. So these things were added. Now we'll see the effects of cytokinin on the plants. What is the use of cytokinin? तो effect cytokinesis. Cytokinesis का meaning होता है cell division, है ना? Karyokinesis and cytokinesis. Promote cell division and it was first discovered and named because of its ability to promote cytokinesis. Cytokinesis को promote करता है, cell division को promote करता है. That's why it is called cytokinin. It produces new leaves and chloroplast in leaves, which is very important function, and enhance lateral growth and adventitious shoot development. तो lateral growth that means side जो branches हैं उनको grow करने में, उनको arise करने में help करेगा. Cytokinins overcome apical dominance. Now हमने auxin क्या था? It was promoting apical dominance. The cytokinins will overcome. That means if we provide cytokinins to the plant, तो it will induce side branches development. Side में भी branches आएंगी, तो उससे क्या होगा? The plant will become dense, thick हो जाएगा plants. जैसे कि हमको boundary करनी होती है plants की help से, hedge कहते हैं जिसको, तो in that case we can provide cytokinins. They delay senescence. Senescence means aging को रोकता है, तो they delay senescence of leaves as they promote nutrient mobilization. Nutrients को transferring from one part of the plant to another. It is done with the help of leaves. So senescence of leaves will be stopped या delay कर दिया जाएगा, so that the leaves remain on the plant for a longer time. Thus DNA suspension can also enhance callus formation in tobacco. Kinetin does not occur naturally. Naturally कभी हमें नहीं मिलेगा. Cytokinin like activities are shown by substances known as zeatin. Zeatin से हमें cytokinin जैसी activity मिलेंगी, which is isolated from corn kernels and coconut milk. So this zeatin can be obtained from coconut milk and corn kernels, which have the activity similar to cytokinins. Some adenine type of cytokinins are zeatin, kinetin and 6-benzyl aminopurine. The ratio of auxin to cytokinin is crucial. तो ये एक बहुत important चीज़ है कि auxin और cytokinin का equal amount में ratio होना चाहिए in plant cultures, as it can be adjusted to promote rooting or shooting. So it will help in both these things. You can see in the figure an increased proportion of cytokinins than auxin in tissue culture is seen to initiate shoot formation in the callus. That means अगर हम auxin से ज़्यादा cytokinins प्रोवाइड करेंगे तो वो शूट फॉर्मेशन में हेल्प करेगा. एंड इफ ऑक्सिन इज मोर, इट विल हेल्प इन रूट